its core, the human body runs on a remarkably simple mechanical trick: the ability to tighten and release. We call the biological hardware responsible for this trick muscle. It is a specialized tissue designed to do exactly one thing: contract and relax. Scale that single cellular motion up, and it drives three major outcomes. It generates the heat that keeps us alive, maintains our posture against gravity, and produces all of our gross physical movements. This creates an immediate biological problem. A human requires wildly different types of motion. We need explosive, immediate power to lift a heavy weight, but we also require the slow, continuous churning that digests a meal. To solve these different mechanical needs, a single generic contracting tissue undergoes radical structural and neurological specialization. If you open a traditional anatomy textbook, it usually attempts to organize these specializations by giving you a series of isolated lists. For example, you'll see a functional list splitting muscles by control, voluntary and involuntary. Directly below that is a separate classification system categorizing them purely by microscopic features, striated versus non-striated. Look closely and you notice a clear overlap. The exact same three muscle types, skeletal, smooth, and cardiac, appear repeatedly under these different headings. Studying these separate lists usually leads straight to confusion. It forces the student to rely on memorization instead of actually understanding the underlying anatomy. We need a different mental model. Instead of reading sequential lists, we should look at how these variables intersect. Biological taxonomy maps the precise intersection of physical structure and neurological wiring. To map human musculature logically, let's build a two-dimensional grid. This matrix charts our overlapping variables. The y-axis represents structural morphology, telling us if the tissue features highly organized cross-striations, which act as a power multiplier, or if it is non-striated. The x-axis tracks neurological control. This plots whether the brain actively, consciously commands the tissue, or if it runs automatically on an involuntary background loop. By plotting any muscle on these two axes, both its microscopic cellular footprint and its macroscopic role in the body become instantly predictable. We will start at the intersection of voluntary control and striated morphology. This quadrant belongs exclusively to skeletal muscle. For a muscle to operate in this space, it needs a specific neurological hookup, somatic nerves. These somatic nerves allow you to activate the tissue consciously, delivering explosive power exactly when you decide to make a movement. Structurally, it requires heavy cross-striations. Those highly organized bands allow the tissue to produce explosive, directed physical force. When we check the anatomical record for skeletal muscle, the parameters match our matrix. It is explicitly defined by those cross-striations and voluntary control. To support this rapid-fire power, the tissue adopts a specific cellular footprint. The fibers form into long, cylindrical, unbranched columns. Because these fibers are so long and demand so much energy, they require multiple nuclei. These nuclei are pushed out to the extreme periphery of the cell to stay out of the way of the contracting fibers. You see this exact profile in the classic examples, such as the biceps or the triceps, and the quadriceps for conscious locomotion. Skeletal muscle's highly organized, multinucleated structure perfectly matches its function as the body's primary consciously controlled force generator. The body also has a physiological need for slow, tireless operations, processes that must run constantly in the background, like digestion and blood flow regulation. To find the tissue that handles this, we look at the opposite intersection: involuntary control combined with a non-striated morphology. This is smooth muscle. Here, the neurological wiring shifts. Control is handed over to autonomic nerves, completely bypassing your conscious thought. The structure shifts alongside it. The explosive power of striations is abandoned. Instead, the biological architecture favors slow, sustained contractions that can maintain tension for long periods without fatigue. The documented cellular characteristics of smooth muscle align perfectly with these specific physiological requirements. Unlike the massive, parallel columns of skeletal tissue, smooth muscle fibers are solitary and spindle-shaped. Because they don't share the same extreme energy demands, they operate efficiently with just a single central nucleus. You will find this tissue operating throughout the internal viscera. It lines the intestinal wall, the uterus, the blood vessels, and the urinary bladder. Smooth muscle trades raw, explosive power for the advantage of tireless, automated internal regulation. Pulling back to view our matrix, 2 quadrants are filled. But this leaves a structural gap, the intersection of involuntary control and striated morphology. This presents a biological paradox. What organ demands the immense, organized physical power of striated muscle, but requires the automated, tireless control of smooth muscle? If you had to consciously remember to beat your own heart, you wouldn't survive your first sleep cycle. But if your heart lacked explosive pumping force, it couldn't push blood against gravity to reach your brain. The solution is cardiac muscle, a highly specialized hybrid tissue localized entirely within the myocardium of the heart. Cardiac morphology borrows from skeletal muscle. It utilizes heavy cross striations to generate the physical force required for continuous pumping. But its neurological wiring borrows from smooth muscle. It relies on autonomic nerves, both sympathetic and parasympathetic, to provide involuntary rhythmic pacing. To make this hybrid system function, the cells undergo a structural adaptation. Unlike the isolated parallel cylinders of skeletal tissue, cardiac fibers branch out, weaving together to connect with one another. Where those branches meet, we find a critical cellular component unique only to cardiac tissue, intercalated discs. These discs serve as an engineering bridge. They allow rhythmic, involuntary electrical signals to pass instantly through those powerful striated fibers. The heart functions by merging these 2 systems, using the strength of striated fibers while maintaining the autonomy of involuntary control. Looking at our finalized matrix, the logic of the system is visible. Skeletal, smooth, and cardiac tissues are precise intersections of morphological structure and neurological routing. We started with the overlapping classification lists found in textbook anatomy. This matrix model provides a framework for these 3 types. If you understand a muscle's role in the body, its microscopic structure and nerve supply follow a predictable pattern. Understanding these classifications reveals how the body's physical requirements dictate its cellular design.